similar interest, maybe you know it. We always been saying that that neurodegenerative disorders involve protein aggregation disorders. This is also a protein aggregation disorder. Alzheimer's disease is a protein aggregation disorder. Parkinson's, Huntington, or disorders of protein aggregations. There are other pathologies also described for the first time 1817, and further this disease was characterized by Charcot in 1800s, and this was a description of this disease, or you can say publication about the disease, an essay on shaking palsy. Motor functions are impaired, that we all know, and so motor dysfunction was described something like cerebral palsy. These are cardinal features, hallmark symptoms: resting tremors, bradykinesia, muscle rigidity. Few more, but I was just saying incidence all across the globe. Primary pathology involves depleted or low levels of or loss of dopaminergic neurons. When I say dopaminergic neuron, you can see the picture on the right. If there are less number of cells, they will make less amount of dopamine. Imbalance of dopamine versus acetylcholine is what is creating this muscular or cerebral motor palsy that we were describing. And it happens primarily in this area called putamen or substantia nigra. Putamen caudate nucleus is the area where dopaminergic neurons are there. Maybe all of you are neuroscience, we all say different regions of the brain are different tissues actually, because they behave very different, chemistry is different. Interestingly, there are non-motor symptoms that you will see in the early phase, and then you will get motor symptoms little late in the time course of the disease development. And we all know probably this disease is a mitochondrial disease. Mitochondrial complex 1 is important. A lot of studies support this transgenic overexpression of PGC. So if we have PGC1 knockout mouse, they are more vulnerable to the toxicity of MPTP, which is a toxin that depletes the cells in the brain. And if you overexpress this PGC1 alpha controls the mitochondrial biogenesis. So number of mitochondria depend on this. Anything that increases or activity of this PGC1 alpha or amount of this PGC1 alpha will be beneficial. It also talks about resveratrol, which also acts on PGC1 alpha and increases mitochondria. So other supporting evidence. In interest of time, I'm not getting into this. This is talking about existing therapy. What are existing therapy? You are supplementing dopamine from outside, fine. But we know there is a problem with that, because your enzymes to metabolize will also be going up. Either you will have to decrease the dose, or another strategy is I can give an inhibitor of monoamine oxidases or other enzymes that are involved in degradation as an adjuvant, so that because peripheral system will also metabolize dopamine. So the kind of therapies it says is L-dopa is one that is given. It talks about dopamine agonist, which will mimic dopamine, monoamine oxidase B inhibitors, then I am involved in degradation of dopamine. Carboxy-O-methyl transferase inhibitors can also be useful. So all these can be possible therapies, and it is talking about the sites or biogenesis or dopaminergic system on the left side. I am not getting into this. So all these existing therapies have adverse effects, because these are all chemical agents. If you look at it, the dopamine agonist, some examples here, then COMT inhibitors, monoamine oxidase inhibitor, sertraline, resgaline, few more, L-dopa we all know, and there are a few more of these types that we can talk of. All right. Adverse effects. So this is just a literature to build the story that there are problems with existing therapies. So we stumbled on this molecule. Compare the molecule on the left, compare the molecule on the right. So it's one amino group is replaced by hydroxyl group. We said let's compare maybe this. At least it would do something to the scheme of things where dopamine is involved. Mainly present in olive oil, virgin olive oil, potent antioxidant activity. and it's been used as cardiovascular protector. So I said this already, structural similarity to some extent, at least the basic backbone was similar. Maybe I can put that down. All right. So key questions were, does hydroxytyrosol have any anti-Parkinsonian targets? Can it rescue dopaminergic neurons? Can it be neuroprotective? Can it affect mitochondrial functions? So the first part was in silico. I will not get into too much details, but you can see we looked at binding of hydroxytyrosol, rasgalene, which is, I said, is monoamine oxidase B inhibitor, other molecules, even dopamine, it does show interactions with monoamine oxidase B. We also did it with monoamine A also and compared that, and look at something very interesting. This is hydroxytyrosol, this is rasgalene. Sites where it binds are almost kind of identical. So that means pocket that is using is also similar. So whenever we talk about this binding, we also do molecular dynamics, which says the complex that is formed is stable, and this just was validating that part. All right. We also look at blood-brain barrier permeability, and then we also look at Lipinski's rule of 5, which says molecular weight less than 500. Other properties, hydrogen donor, acceptor properties, all fits into the criteria that we are interested. It is also blood-brain barrier permeable also, which also supports this hypothesis that maybe there won't be any issues of crossing blood-brain barrier. So I'll just say that maybe whatever I said is already explained. I will skip this also. So now we wanted to validate whether it will do something in vitro or in a real situation. First part was in silico. So I would say why we started doing in silico. I never did in silico. This was a shift we had during COVID time because labs were closed. So we all decided let's do something based on in silico. So what we did was we wanted to look at whether it will work in an actual scenario. So we took platelets which is rich in monoamine oxidase B and then screened this enzyme activity. We also looked at whether it will have any effect on synuclein polymerization also using thioflavin T fibrillar acids. So this is the concept we are saying, platelet isolated, look at monoamine oxidase B inhibitor reactions. And maybe I'll just take a call, something, give me a second. Hello. So this is the kinetics you can see, compare hydroxytyrosol versus rasgalene. So IC50 is 4.82 nanometers. For hydroxytyrosol it was in micromolar concentration, higher, but this is a phytochemical, that's a synthetic drug, there has to be differences, but it does inhibit monoamine oxidase. That means it can be inhibitor of monoamine oxidase B. Even synuclein aggregation, you can see hydroxytyrosol. This is synuclein aggregates. When you treat it with hydroxytyrosol ratio 1:4, you can see aggregation is, this is TEM scan, and the top is the fluorescence signals to pick up where it says aggregation is prevented when the ratio of hydroxytyrosol to synuclein was 1:4. So two sites where we could show that it does inhibit in vitro. So we wanted to take these studies to in vivo system two models we used, C. elegans and MPTP mice model. Why C. elegans? I will just talk about it briefly. Certain number of neurons, 308 precisely, and you can map all these neurons. Dopaminergic neurons 8, all can be seen individually because optical clarity of the worm is also there, and you can use these mutants. N2 is a wild type mutant. NL5901 is a mutant where you have the fluorescent protein, yeast fluorescent protein being expressed in the muscles. I'm sorry. And BY555 is a mutant that has GFP in dopamine containing neuronal stroma and processes. So I can see dopaminergic neurons in this C. elegans. CW152, which doesn't have complex one. So one model which is a mutant of PD, this model is where GFP is being expressed in the muscle. There is another model where you are expressing a protein in the muscles, and this is a wild type mutant. So we wanted to see whether there is any toxicity of hydroxytyrosol. No toxicity at all at higher concentrations up to something like 500 micromolar. And this is synuclein aggregation assay. This is done in the strain called NL5901, where we said this fluorescent protein is being expressed in muscles, and this is synuclein protein. What you can see is this is control, higher fluorescence. I will just talk about where the significant difference is. You can see green fluorescent is gone down, indicating that it prevents aggregation in the muscles, which is an evidence in favor of hydroxytyrosol being useful. So likewise, yeah, that's just highlighting the bar. So other thing that we want to tell you is monoamine oxidase is also an enzyme required for activated converting MPTP, which is a toxin, to its active form, that is MPP. This enzyme is present in astrocytes primarily. So when you inject MPTP into rats, this has to get converted to MPP, which is neurotoxin actually, and will kill those neurons. All right, this is just a background. So you will, this strain which has bright GFP fluorescence coming from dopamine containing neurons, if I treat them with MPP, it should kill those neurons. Am I right? Because I am only seeing bright fluorescence from dopaminergic neuron. I treat it with MPP. What you should see is, all right, this is control, strong fluorescence from the dopaminergic neuron goes down, recovery with hydroxytyrosol, although not 100%, but significant recovery after hydroxytyrosol. So it's rescuing dopaminergic neurons from MPP induced toxicity. So another test that is done is called neuronal repulsion time. Repulsion time is linked with content of dopamine. Repulsion time, so you give this chemical, the worms would repel because this is an irritant for the worms. So this compound nonenal, once you give, what you see is this is the controls. This is the repulsion time. I give MPP, there will be loss of dopaminergic neurons, repulsion time has increased, and with hydroxytyrosol this time has gone down. That means dopamine containing neurons, functional assay of dopamine in the C. elegans. This is done using NL5901 strain and BY555 strains. So I can also look at mitochondrial viability with the strain that was GAS1 mutant. So GAS1 mutant is where the mutant did not have complex 1. So you can see activity is low, hydroxytyrosol mitochondrial fluorescence is higher in these cells because mitochondria has been seen using mitotracker deep red staining in this cell. So I am just not getting into all the data for this, maybe we can skip some, and then we also wanted to look at what happens to the expression of these complex or mitochondrial genes that will be involved in PD also. So we looked at PDR1, LAGR1, PINK1, ERK1, G, SRK1 in this NL5901 strain. You can see the activity has gone up in one which are beneficial, gone down in the case which are not good. So maybe just summary of what I will say. The mRNA expression shows hydroxytyrosol reduced synuclein toxicity in the strain of C. elegans by upregulating expression of PDR1, LAGR1 and downregulated expression of GRK1. Rest of the things I already discussed with you. We also wanted to go to in vivo model, the rat model, because sometimes it is helpful. Use different doses of MPTP, pre-treatment, post-treatment with a hydroxytyrosol. The dose of MPTP we used was 20 milligrams per kilogram body weight, 4 injections in a single day. That's what is the accepted dose for creating this model. Something important, we have to use here C57 black 6s. We have a data where we compare two strains of mice and we say this C57 black 6 is the only relevant model. So one of the behavior tests for seeing whether they develop PD is catalepsy, which they are asked to made to walk on a bar, and MPTP treated animals day 16. You can see the descent latency is increased significantly, indicating motor function is impaired. Narrow beam walk test, maybe interest of time, I will say you challenge the animal to walk on a narrow beam, which wider at one end gets narrow at the other end. So it's a challenge for them to walk if their motor functions are impaired, and the one end is baited, and you can see there is time to cross the beam is increased. Day 16, hydroxytyrosol has been beneficial in them also, and probably we did more of behavior test, and one of them is pole test, and I'll not get into data because I was told we can do a quicker session on this. Maybe if anybody wants, we can discuss that. Rotor rod also, probably many of you have done this test. One test we do in our lab for motor function is called footprint analysis. Normal animals would have hind and forelimbs overlap. MPTP, you can see this overlap is problematic. Post pre-treatment beneficial, they come back to the same gait is restored in the animals. All right. Neurotransmitter levels. I'll be interested in few things that I'd like to talk to you. Dopamine depletion, restoration, hydroxytyrosol not detectable in these tissues which have not been given. You can see hydroxytyrosol in the brains once you treat that. You can estimate hydroxytyrosol in the brain also, and significant increase in these animals. We can also look at monoamine oxidase activity. It is suppressed in the treated animals, indicating it also does inhibit the activity also. So anyway, so you can also look at dopaminergic neurons by tyrosine hydroxylase staining, and immunohistochemistry reveals there is a reduction in the brain, restoration. This is reduced tyrosine hydroxylase, restoration in the brains after hydroxytyrosol treatment. So I will not get into repetitive data. This is quantification of the tyrosine hydroxylase dopamine transporters, indicating the same kind of findings that it does restore dopaminergic neurons. Nissl stain. Then we also looked at mitochondrial functions. They are restored after giving hydroxytyrosol. This is done using oxygraph, where you look at oxygen consumption and then you can look at different complexes of mitochondria. This is the data from there. You can also look at NRF2 protein, which is an antioxidant response gene, and we can see if I just compare MPTP and post hydroxytyrosol you can see significant difference at the level of western blot also. Mitochondria as well, MPTP toxicity, they are restored. Electron microscopy also says something similar. All right, mitochondrial biogenesis genes, PGC1 alpha, suppressed significantly following MPTP exposure. Recovery significant happens after other genes like NRF1, which is also involved in mitochondrial biogenesis. TFM also shows the same trends. So almost similar kind of things. This is again western blot for some of these genes in the same order. We can also look at mitochondrial dynamics. I'm not getting into that. So we can say it favors. So if I have to sum up what all we could say based on our study, we could say hydroxytyrosol prevents synuclein aggregation, it prevents degradation of dopamine, antioxidant also functions, mitochondrial functions, and all this would contribute towards having beneficial effects in PD. So we are not saying this is a treatment, we are saying still this is a lead molecule. You can improvise this molecule or you can use this molecule as an adjunct to existing therapies because it has pleiotropic properties, antioxidant, mitochondrial biogenesis, few other properties. So if we have to say treatment, we still have to work on this molecule more, and this is the work primarily being done by Anjana, who was a PhD student and now works for a company in Bangalore. And maybe since I have slides, I forgot to talk about this. I also represent one of the entities on the campus called Northern Region SNT Cluster. You might have seen this logo also in our certificates. It's called PI Rahi, Punjab University, IIT Ropar, Regional Accelerator for Holistic Innovation. Why I wanted to talk about this is, we all the time should not be interested in publication, maybe think of something which is innovative, which could translate into something real. So we say regional SNT cluster, we have a mandate of this region of India, J&K, Himachal, Punjab, Haryana and Chandigarh also. And it is a section 8 company, that means we are out of the bureaucracy of Punjab University. Punjab University and IIT Ropar are 50-50 stakeholders into the company. These are our thematic areas. Anybody having an interest can come back to us. Agriculture, food processing, pharma, healthcare, medical devices, any indigenous technology, development and optimization, sustainable mobility, green energy, waste management and waste from wealth. So we do support startups, we accelerate startups, anything which is innovative, which can be a regional solution provider, which can help this region. So we have a lot of programs that we launch, we do capacity building. So anything under science and technology umbrella is part of it. We are the 7th cluster in the country. All others are city-based clusters. The recent addition cluster is an interesting addition, and I probably think that maybe you've not heard that name, maybe explore that. AMTZ, Andhra Pradesh Medical Technology Zone, which is a huge space, 270 acres, you can say sector 14 campus of Punjab University, could be similar space where they only talk about medical devices coming from that area. So look at the kind of technologies they come up. Delhi has a cluster called Drive, Pune has a cluster called PKC, Bangalore has a cluster, Bhubaneswar has a cluster. So we are the 7th cluster, and this idea of cluster also came from Chandigarh. We had an entity which Dr. Gurjasprit represents called CRIC, but CRIC, we are now an official, you can say, and legal entity which got recognition in the form of cluster and recognized by principal scientific advisor to Government of India. That means our reporting is to the Prime Minister's office. And thank you for your time and attention. I did discuss short because of what the thought was. Anybody with a question, I'd like to answer that. So I just wanted to give a flavor of what we do. We keep on evolving in terms of our interest, and we do multiple different areas of research. You also, I think,
If I buy from the company, very expensive. Customized, these are customized molecules. That's a good approach. But better is what you said, phytochemicals advantage. So if you define the target, that is easier. If your target is not defined, then the approach becomes very difficult. So I also had an interest that I know I want to pick up a disease, not define a target, and see where I should have been happy if my system tells me what is the likely target of this molecule. But there is can do the matchmaking. Anybody else have a question? Yes.
What is the mechanism of action of this hydroxyl? So you said it is helping in improving So also you said that it has a structure similar to that of dopamine. So is it competing with dopamine to interact with the different receptors?
That's what we think. That it is. So it has multiple targets. It itself is antioxidant, which I do not highlight in my study. So we started with looking at if dopamine is what is the target. But if you look at literature, it says it also targets mitochondrial biogenesis. And even I would say, if you look at more recent literature, people may have found more targets of this molecule. I said, if you look at initially when I said it is a cardioprotective molecule, some literature suggested it is beneficial for mitochondria also. It has good effects on biogenesis of mitochondria. All kinds of not known. So pleiotropic properties are something that I will say. But one target is definitely monoamine oxidase B, but there are other targets also, which could be secondary or primary that we cannot say.
So for in-silico studies, what would be targeted in
What we used monoamine oxidase B from humans. So in crystallized structure is required. I think you probably, maybe Dr. Dingma did talk about that. So you need a crystallized structure that makes life easier. If you don't have, then it is a bigger challenge. You will have to then think of some other approaches to find it. Or you can predict the structure. Probably there are alpha-fold site probably that can predict the structures of the molecule. Probably can use it. I'm not having that much expertise in silico work, but we collaborate with somebody from BHU who has high-end computing facility. Because for doing molecular dynamics, you need high-end computing facility. If you don't have it, then probably there is a challenge in having good results. So they have a bunch of supercomputer there in BHU that they use. We also have a supercomputer on the campus, but probably we are not using them for in-silico.
Sir, in silico, like we have done previously, like we are only limited to know the admit profile of inhibitor if we know the late compound address. Nothing more in silico other than admit profile.
You can know a lot of things. Even the enzymes that will metabolize it, there are n number of properties you can examine from. Not from exact precisely admit, but there are other tools along with that which will give you more information on pharmacodynamics of the molecule also. So more can be done. I think Dr. Nidima is the best person probably in the campus who can tell you all those other tools that can be used. Maybe we can also connect you with person with whom we collaborate, maybe tell you all other tools that can be used. I also kept my in silico work very precise because the meaning I wanted to convey that wanted to give a flavor of it rather than deeper into each part of the segment. I didn't show all the analysis that we do for in-cell. There are more tools we can use. So one name in a thesis can be in-cell also. So it tells you, give you some leads. Anybody else with a question? All right. Thank you. No more questions?